My name is Thomas Martinez, and I have been funded by the Anti-Cancer Challenge Pilot Foundation. The research I'm about to tell you about is a very nascent field, nascent idea. Research in my lab is focused on identifying new genes in our DNA, in our human genome. These are genes that have been there the whole time, but have been previously overlooked. And the hope is that these new genes can actually be important for regulating the phenotypes we see in cancer that drive its growth, that make it malignant, that drive its migration potential. Then maybe we can develop new drugs to block them. A secondary goal we have, which is just as important, is that maybe some of these new genes will produce proteins that can instead also serve as markers for diagnosis. It turns out that you can have proteins that are less than 100 units in length. They can be very small, and we call them microproteins. And so the main questions driving my research in my lab are how many microproteins are encoded in our genome and what are their functions? And in this case, it's about cancer. We thought this would be a wonderful opportunity to look for microproteins in pancreatic cancer because there's a dire need for new ideas for treatments and new diagnostic markers in pancreatic cancer. One of the helpful things, at least, about pancreatic cancer is that we understand its genetics that cause it fairly clearly. It's mostly driven by mutations in 4 genes, the most important of which is this KRAS gene here. And what we found was astounding. In these cell models from these pancreatic tumors from these mice where we turn on KRAS, in total we can identify over 10,000 microproteins. So we know tumor cells can also let go and release a bunch of proteins as well because they may be talking to other cells in the body or other cancer cells. And if we can identify microproteins that are also being released by these tumors, we may be able to detect them in blood and use that as a means to identify the tumor in a patient sooner, which will help clinicians. Identifying new microproteins that can regulate growth in the pancreatic cancer or new microproteins that can be released by the pancreatic cancer cells. We're hoping to both be able to tackle and identify new targets that we can develop therapies for, as well as new diagnostics. This is still very early days, but like I mentioned, this is a very kind of fringe, nascent field for cancer research. And so for something as deadly as pancreatic cancer, we think it's worth the effort to try this kind of out-of-the-box thinking.